The intestine also secretes water and electrolytes into the lumen so that digestion and absorption can occur effectively. This image summarizes the daily fluid intake, which is about 2 liters, and the volume of secretions that enter the GI tract from each source, about 7 liters in total. So of this approximately 9 liters of fluid that enter the GI tract each day, almost all of it is absorbed in the small intestine. Some is absorbed in the colon, and only very little, about 100 milliliters, is lost in the feces each day. Most fluid absorption occurs specifically in the proximal small intestine by the paracellular route, where the tight junctions that link the enterocytes are leakier. The intestinal epithelium contains absorptive cells as well as secretory cells. The intestinal mucosa is folded and contains many protruding villi as well as invaginations called crypts. This single layer of epithelial cells originates from stem cells in the base of the crypt, and they migrate upward towards the tip of the villus, where they undergo apoptosis and are sloughed off into the lumen of the intestine. As these epithelial cells move up the crypt-villus axis, they change their characteristics. So while the cells are in the crypt, they're generally secretory, and they have many CFTR, or cystic fibrosis transmembrane regulator channels, that allow secretion of chloride ions into the lumen. Sodium and water follow paracellularly, and the result is secretion into the lumen. But as cells move up the crypt and onto the villus, they're transformed into net absorptive cells, and they have many transport proteins that allow absorption of nutrients from the lumen into the blood, some by secondary active transport coupled to sodium, some by facilitated diffusion. So the crypt cells generally secrete water and electrolyte, and that's driven by the cystic fibrosis transmembrane regulator channel, or CFTR, while the villus cells generally absorb water and nutrients, and that's driven by several transporters specific for each type of nutrient. If we focus on absorption in the cells of the villus first, we can look at the isoosmotic absorption of water. So there's solute that is absorbed from the lumen, and then water follows paracellularly. This mechanism is similar to the proximal tubule in the nephron. In the intestine, there are several distinct sodium amino acid cotransporters, each one specific for a different class of amino acids, and they're responsible for the sodium-coupled absorption of amino acids. There's also a sodium glucose cotransporter, and this is responsible for glucose uptake across the luminal membrane, and both of these are examples of secondary active transport. So it's the sodium-potassium ATPase on the basolateral membrane that keeps the sodium concentration low inside the cell, and that sodium gradient is used to couple the downhill movement of sodium to the uphill movement of glucose or an amino acid. This overall uptake of sodium increases the intracellular sodium concentration, which increases sodium absorption across the basolateral membrane by the sodium-potassium ATPase. In addition, there's a sodium-hydrogen exchanger that drives secretion of hydrogen ions in exchange for sodium ions. The source of these hydrogen ions is intracellular carbon dioxide and water, which are converted to hydrogen ions and bicarbonate in the presence of the enzyme carbonic anhydrase. The hydrogen ion is secreted by the sodium-hydrogen exchanger, while the bicarbonate can also be secreted by a chloride bicarbonate exchanger. The net result here is the absorption of sodium and chloride into the cell, which can then be absorbed across the basolateral membrane. This absorption of ions and nutrients from the lumen creates a large osmotic gradient, allowing water to move by osmosis primarily through the paracellular pathway, through leaky tight junctions between epithelial cells. So the net result here is isoosmotic fluid absorption. We may normally think of the small intestine as primarily absorbing water and nutrients, but if it only absorbed fluid, then it would quickly become dehydrated. To maintain a fluid lumen, a constant secretory process needs to occur, and this happens in the crypt cells and is driven mainly by chloride movement out of the cells and into the lumen. Chloride's transported through this CFTR, while sodium and water move through the paracellular pathway. When they haven't been stimulated, crypt cells secrete little chloride, and that's because these chloride channels on the luminal membrane are closed. But upon stimulation by bacterial enterotoxins or hormones or neurotransmitters, these chloride channels open so that the chloride can be secreted. Chloride is brought into these cells across the basolateral membrane by a sodium-potassium 2-chloride cotransporter, similar to the one found in the thick ascending limb of the loop of Henle. That chloride then moves through these open chloride channels on the luminal membrane and into the lumen. Sodium passively follows chloride secretion, and water follows the sodium chloride into the lumen. Binding of acetylcholine, or VIP, to the basolateral receptors causes activation of adenylyl cyclase. This results in the generation of cyclic AMP, and cyclic AMP acts to open the chloride channels in the apical membrane, allowing chloride secretion. Sodium and water then follow the chloride into the lumen. This process maintains the fluidity in the lumen by replacing the fluid that has been absorbed by the villus. So under normal circumstances, there's a balance between the secretion by the crypt cells and the absorption by the villus cells. However, in the presence of cholera toxin, for example, there's dysregulation of ion and water secretion, where the secretory process becomes excessive and exceeds the absorbed volume, resulting in secretory diarrhea and severe dehydration. Cholera toxin is able to irreversibly bind and activate the G protein coupled to the adenylyl cyclase, which increases the intracellular cyclic AMP and maintains an open chloride channel as long as the cholera toxin is present. This results in intense secretion of chloride, with sodium and water following as long as the chloride toxin is present in the lumen.